Hello again, Bio 111 students, and welcome to week 10, chapter 11 this time. And today we're discussing meiosis. And meiosis is a very important part of how we accomplish sexual reproduction. So let's define a few terms first. Sexual reproduction is when a set of parents we'll say this is a cell from each parent. A set of parents gets together and through meiosis produces gametes. So we have 46 chromosome cell up here in a human, 46 over here, the female, and we have 223s, and in the end we end up with 4 gametes produced from each original cell that went through meiosis. So we end up with haploid gametes, and they will fuse together during sexual reproduction to produce one diploid offspring. So you have one of these guys, one of these guys, fuse together, and they make this 46 chromosome individual cell, which is an offspring cell that will then undergo many divisions before it becomes an embryo. So we've to define that. Now we should define the word gametes. So in humans, gametes are haploid, sperm, and eggs. And the word haploid, we always say that. Just remember that it has half the normal amount of chromosomes that a normal cell would have, and when they get together, they produce a cell that has the full 46 chromosomes. So in humans, sperm is one of the gametes, and eggs are the others. So, genes are the unit of heredity, and it is a sequence of bases, and each is a unique genetic code for a particular protein. So we say genes, code for proteins, and what we're going to have is different combinations of the following letters, which are the names of the bases, and that's adenine, thymine, glycine, and cytosine. So a locus is the place on a chromosome where we will find a particular gene. So let's say it's right here. That would be a locus. So a place on a chromosome where a particular gene is found. So an allele is one of, so when we talk about alleles, you'll remember this from going over our human genetics lab that we just did. We talk about something like A and A. Let's say this is a heterozygous pair of alleles. and they're found in a particular place. And so an allele is one of two gene variants. This is one gene variant that's dominant, and this is a gene variant that's recessive, that arise by mutation, and they're found on the same place on a chromosome. A mutation is a change in the sequence of DNA, and it can be harmful, it can be beneficial, or it can be neither. It can be neutral as well. So let's say we have a set of genes here, and we have A, T, C, G, T, A, G, and if we remove one of those, that's called a point mutation. So then you end up with those two getting together, and you have CAG instead of what you had before, and that's called a point mutation. So it's a change in the sequence by one thing being changed. So let's say that T became an A. That would be a point mutation. If it was completely removed, it would be called a deletion mutation. So those are two types of mutation. You don't really have to know the types of mutation, but I wanted you to know that there are different types of mutation. You can also have a whole sequence of stuff get wiped out, and so it will join again up here. And who knows what the sequence will be, but it could be G, C, repeating, and then AT as the new sequence. And the reason why this could be harmful is because it could change the form of particular proteins, and because proteins fold differently, it could result in a structure being harmed, and that structure may not be able to do its particular function. We might see something like this with sickle cells that we talked about earlier. Sickle cells that are found in some people, they're unable to perform the functions of normal blood cells, red blood cells, and they're unable to carry oxygen. So that would be a very serious problem if a genetic defect caused that. There are also cancers, another form of problem that you get with genetic mutations. But sometimes they can be beneficial, meaning that randomly genes get changed, and a mutation happens that leads somebody to have maybe something like slightly more hair in an animal that's found in a cold environment, and that would be a beneficial mutation, and then that mutation may be passed on because that animal would be more genetically fit for their environment than the other animals. So and sometimes it's neutral. Sometimes the mutation takes place in a part of DNA that really doesn't code for anything too important, so we never know that the mutation happened, and it's neutral. So let's define the terms diploid and haploid. So let's say we have a diploid set of chromosomes, and let's say this animal has 4 sets of chromosomes. Okay, so this animal, diploid, its diploid number is 8, because you see 1, 2, 3, 4, 8. So 2n is diploid equals 8, and its sperm and egg cells will be the only cells in its body that will be haploid. Diploid is all body cells except sperm and egg cells. So the haploid number for this one, the chromosomes will look like this, because some of them will be split away during the process of meiosis. So the diploid number is simply n, and that will be equal to 4. So sometimes we get a situation where we have polyploid. So that is called a 2n plus situation, meaning that instead of a pair of chromosomes, you could end up with 3 together, or 4 together, or 5 together. That is called polyploid. So this situation, I would draw 3 together. That's polyploid because it's over 2, but it could be any number. And this is a situation that occurs. So, an autosome is any of the numbered chromosomes. So when we see a chart of chromosomes, like we saw in our lab, we see 23 space for 23 pairs, and all of them except the last set of chromosomes are autosomes. The sex chromosomes make up the last pair of the 23 pairs. So we see when we're talking about autosomal dominant and autosomal recessive, we're talking about the trait being on the autosomal genes, so the autosomal chromosomes, which means not the sex chromosomes. So X-linked, that is not autosomal, that's X-linked. And X-linked refers to the sex chromosomes, which are the X and Y chromosomes. And their job is to determine the sex of the offspring. That's their only role. So we have stages of meiotic cell division, and we're going to talk about that now. Meiosis stages. So you are going to hear some similar words that you've heard before when we talked about mitosis. And it will take that PMAT format. And that is prophase, metaphase, anaphase, telophase. And they have those in common, but you'll hear those used twice in meiosis because we have meiosis 1 and meiosis 2. Meiosis 1 is not like mitosis, but meiosis 2 is a lot like mitosis. So we'll see, before we do anything like that, we'll see interphase. And interphase isn't talked about a lot in the book, but it is a very important phase, because during the interphase, that's when we have a replication of DNA in order to get ready for cell division. And so it's always the very start. And that happens in particular in the S phase of the interphase. So that's an important thing to know. So we talked about PMAT as a way to remember the order. And the very first one of those is prophase. And so meiosis 1, and we have prophase 1, because we will have second prophase. So in prophase 1, uh, within the nucleus, the chromosomes start to condense and become visible. So we have them start to condense and become visible. And we also have the centrioles start to develop spindle fibers through microtubules. So microtubules start to develop, and they're beginning to form the spindle fibers that we'll see later. And one of the things that's very interesting about prophase 1 that we didn't see in mitosis is that we start to see homologous pairs of chromosomes. Now, the word homologous means that the pair, each chromosome in that pair codes for very similar things, and they're similar in size. So we'll see homologous pairs begin to get together. And what's really important about this is we might start to see, well, we will start to see some crossing over. And just so you can see this bigger, I'll give you an idea of what crossing over looks like. So this is one pair, and it's here, and another pair of chromosomes come along and crosses over. It's kind of like a lazy flopping over, or whatever you want to call it, and as a result, some of the genetic material on this chromosome gets deposited on this one, and there's a swap of information, you might call it, but you get a swap of DNA between the two chromosomes during crossing over. Prophase 1 is the only time that crossing over occurs, but crossing over is important because it leads to an increase in genetic diversity, because these two guys are now not the same as they were before. They have different DNA. So here we have prophase 1, and we also start seeing the nuclear envelope coming apart, and what we'd expect to see in a prophase in meiosis. So now we get metaphase 1. And there are similarities between metaphase 1 and mitosis, but there are also some differences. And you can probably guess the similarity is, just like I said, meta you can think of as being middle, and so you get stuff lining up in the middle. But instead of it being a single line of chromosomes down, we have two lines of chromosomes, because they paired up. And we have kinetochores beginning to form on the chromosomes. And we also have spindle fibers. Going back to the centrioles. Only this time, each spindle fiber, instead of pulling on a single sister chromatid, it's pulling on a full chromosome, full set of full pair, a full chromosome, I should say, and so a pair of sister chromatids, and it's pulling those away. And so in anaphase 1, we'll put it up here. Instead of a pair of sister chromatids being pulled apart, we're seeing this. And over here we're seeing the same thing. And we're seeing a nuclear envelope being reformed. Around each one of these. And then we go through cytokinesis, and that basically finishes the process of giving us two separate daughter cells. Each one of these is a haploid daughter cell. So we are not done with meiosis yet. There will be a whole new set of these processes happening. So we have cytokinesis splitting those two cells, and then we start to see a new set of meiotic division happening, and this is called meiosis 2. So, again, we start to see a second prophase, and we call this prophase 2. And this looks a lot like mitosis, because what we have in prophase 2 is chromosomes condense, meaning they become visible, and the beginnings of spindle fibers beginning to form. And the reason why I say it looks a lot like mitosis is because these spindle fibers, when we get to metaphase 2, line up in a single line right down the middle of the cell after the nuclear membrane has disappeared, like we would expect to see in the prophase. It starts to come apart, and the spindle fibers this time do pull on each sister chromatid at the kinetic core, and this time in anaphase we are going to see what we'd see in mitosis. So this is going to be anaphase 2. Anaphase 2, and we get chromatids being pulled apart and similarly we'll start to see a splitting happening. One thing I do want to say is there is a telophase, and when I went earlier on I said anaphase 1. There's a telophase that follows that, and it finishes up the process before cytokinesis happens. Basically the telophase is where you've got the complete split of the two nuclei, and they start to form a new nucleus, and cytokinesis completes the process. So we have anaphase 2, sister chromatids pulled apart. Then we have telophase, where we start to see the formation of two nuclei, and then cytokinesis, and that would be telophase 2. Cytokinesis, where we start to see the reformation, and we end up with a total of 4 new haploid cells. So, for instance, from each parent in spermatogenesis, each cell that undergoes meiosis leads to 4 separate sperm cells. And one of the things that I want you to know is that the resulting cells are not identical, so it's not like mitosis in that way. And the reason for that is because we have independent assortment of genes going on, like what genes go where is independently assorted, which we don't see in mitosis. And also we have the crossing over in prophase 1. We don't see that in mitosis either. So we've covered the major events of meiosis. And now let's talk about random mutation. We talked about that, where we have deletion or insertion of parts of genetic code. And that ends up promoting genetic diversity. And there can be good, bad, and neutral effects of that. So we talked about deletion. We could also talk about insertion. So let's say this piece here, and it's ATG, and it gets inserted into our DNA at this point, and we have so that will change things a little bit, and you have T, your A, T, G, and so on down the line. So that will be an insertion mutation. So we talked about crossing over, and it happens in prophase 1, and it happens between homologous chromosomes. The shuffling of chromosomes is an interesting concept. I want to talk about that. So during this prophase 1, when we start seeing the chromosomes becoming visible, it can be quite random how they end up together, and homologous chromosomes can encounter each other in a very random way, and as a result of that randomness, it's called shuffling. So it's almost like shuffling a deck of cards. What's going to come out of it can be very random, and that also is a source of genetic diversity. And that is why, even though you might be recognized by your neighbors as being sisters or brothers, you're not genetically identical. You don't look like each other, and all of those random shuffles and crossing over mean that although you're closely related, people can still tell you apart. And one of the other things that happens that's a very random event is millions of these guys all take off swimming towards one of these guys. So we have an egg over here, and you have a million sperm heading for the egg, and again, that's one of those very random events. Which set of gametes are going to end up fusing? It's a very random event. All of her eggs are not identical. All of his sperm are not identical. So one individual comes out, the fusion of gametes, is a random event. And because it's such a random event, we don't know which pair are going to get together. It also leads to genetic diversity. So, again, it's why you don't look like your sister or your brother unless you're genetically identical twins. So, thank you. That's all I have. That's all I have for this week. Have a nice weekend. Thank you. Goodbye.